We are recording. All right, let's see. Is that our key back there? Or Dawson?
Awesome.
All right. See, bars is here. WBSU here. If I remember correctly, Bonner is online. Yes, Bonner, Castro. All right, so Bonner is online again. If you're online, you have to send those words before 9 a.m. to be considered present. Clay is not here. Dawson's here. Ellis is here. Paula is online. Green's not here yet, I don't think. Harrison is here, WBSU here. Jensen is here, WBSU here. James is here. You ran WBSU stuff? It's hard to see, okay. Kesterson won't be here today. King isn't here yet, but he's online. Lewis is here at WBSU here. Linda Q is back there. You ran WBSU stuff? Yep. WBSU here. Morrison is here. Okay. Couldn't tell, that hat almost looked like it was a state hat. I think Grace is online, maybe? Anyway, Ward is back there. Wilcox is not here yet. Wilder is not here yet. Parks, it's not here. Back to the top. Aki, it's not here. Boggs is here. Bonner's online. Clay's not.
I am here.
Oh, what was it?
I am here. You said I wasn't here.
Oh, okay. Bonner's online. Clay, must be online or something. Nope. Clay's not here. Dawson is here. Ellis is here. Paula's online. Green, not here. Harrison's here. Vincent's here. James is here. Kesterson's not here. King is online. Lewis is here. Linda Q is here. Morrison is here. Grace is not here. Ward is here. Wilcox is here now, right? Yep. Wilcox is here. Tyler is online. Don't forget to send the words before 9 a.m. Oh, it's 8:01. Running behind. Parks is not here yet. All right. Really well, yeah, we're ahead of the game, so I'll spend a little bit of time. Really quickly, let me remind you, the lab we're doing today, we have to do lab report. So let's see if I can remember. I'm gonna show you where to find the lab report, if I can remember correctly. Well, first of all, now this is just the quickest, easiest way. If you go here to the announcements, which you guys should be reading the announcements anyway. I have a feeling most of you aren't. And now I'll just go ahead and tell you why I've been hinting at it for half the semester. A lot of times in the announcements, there's a sentence that says, if you read this, reply back to this for extra credit. So, read all the announcements. There's hidden extra credit there for you, and obviously they're important. But anyway, if you go here to the photo respiration lab, everything you need should be here, right? So we've got the video from the pre-lab lecture. If you forgot anything I said, you guys can still get extra credit. For those of you who are here, you can still watch the video and answer the questions for extra credit because it was the same lecture, but I probably added some information to that lecture that I didn't say here and vice versa. What else? Here we go. Pre-lab, we all know about that. Good job on the pre-labs, everybody. Here it is. So this is the lab report. When you find the lab report, this is where you'll find it, right? Yeah, that's it. And then to turn it in, let me see here. If you go to list of due dates, and then scroll down to the lab report. I can see it. Pre-lab, where you at? Pre-lab. There we go, lab report. So first of all, there's the due date, right? October 18th on a Friday. Click here, and then it'll take you right there to where you submit it. Any questions? All right. And the lab report is not an exam, right? It's not a quiz. And what I mean by all that is you don't have to do it alone. So if you need my help, let me know. I can meet you online and work on it with you paragraph by paragraph. I'm here to help you. All right, so are there any questions about lab stuff? Make sure you're here by 10. We're gonna get started immediately because we already did the pre-lab lecture last week. And obviously this lab has a lot of waiting. So the quicker we get started, the quicker you'll get out of here. All right, all that out of the way, let's get back into this lecture. First word for attendance will be egg. Right there, first word for attendance, egg. Anyway, this is where we left off. Prior to this slide, we were talking about how there's asexual reproduction, which is mitosis. And then we said there's also sexual reproduction, which is done by meiosis. And it's the opposite of asexual reproduction, right? So asexual reproduction doesn't use egg and sperm. Sexual reproduction does. Obviously, since asexual reproduction doesn't use egg or sperm, then we don't have to worry about the production of egg and sperm. But as far as we're concerned in this class, and we'll cover it next chapter, that's mostly what we're going to focus on is that production of egg and sperm. We're not even going to talk about, we'll mention it, but we're really not going to get into what happens after the egg meets the sperm, right? We're focused on how we make those eggs and how we make the sperms. And again, that'll be next chapter. Now we'll point this out. The last textbook combined these two. So sexual reproduction and asexual reproduction were on the same chapter. And I'm kind of sticking with that theme, which is why I'm mentioning sexual reproduction when we technically haven't gotten to that chapter yet. So again, a little review before we jump into the specifics. There are two types of cell division
in the lives of sexually reproducing organisms. So we're talking about organisms like us, not the asexually reproducing organisms. But for sexually reproducing organisms, we have mitosis, which you guys are doing right now. As you sit here, you are going through mitosis. You are reproducing at the cellular level. And then, of course, there's meiosis. And meiosis is used for what we usually consider reproduction. So if you remember last week or Monday, I said, how many of you reproduced? And only Bree was the only one to answer, raise her hand, because she's the only one apparently who has a child in this classroom. So she raised her hand. But remember, that's not the only kind of reproduction we're talking about, right? But as far as making a new organism, yes, that is what meiosis is used for. And again, we'll talk about that technically in the next chapter. There's a really stupid way you can remember the difference, because you definitely do need to know that mitosis is for asexual reproduction and meiosis is for sexual reproduction. This is really stupid, but I learned it in undergrad, and it's a little bit inappropriate, so forgive me if it offends you, but it might be a way to remember it. So mitosis, right, when you're having sex, you don't use your toes, right? I mean, you might if you're in the feet, but if you're having sex for reproduction, then toes are not the things you're going to be using to reproduce another organism, right? So that's how I always remembered that mitosis was not for sexual reproduction. Anyway, let's jump into it. Now we're going to jump into the second main bullet point of this chapter. I'm going to try to slow it down because we're a little bit ahead of the game. But we're going to talk about the cell cycle and mitosis, right? So let's jump into the cell cycle. Before we can talk about mitosis, we have to talk about the cell cycle. And before we can even talk about the cell cycle, there's some other background information that you need to know, and that's what we're going to talk about here. And that first bit of information that you need to know is about eukaryotic chromosomes. And the good news is you're already a little bit familiar with eukaryotic chromosomes, because in chapter 2 or 3, I think it was 2, we talked about biological molecules, right? And we talked about nucleic acids, right? We talked about DNA and RNA. So you've already been introduced to that concept in that chapter. And then later, we had a whole chapter on how cells work, right? So in that chapter, we also talked about chromosomes and the nucleus and things like that. So some of this will be a review. We're going to kind of skim over what we've already talked about, and then we're going to start diving a little bit deeper. So let's talk about eukaryotic chromosomes. Also, I want to point out, this is obviously, it's right here in front of you. We are focusing on eukaryotes, right? We don't even talk about prokaryotes, which are like bacteria. So everything I teach you, in the back of your mind, you should think to yourself, well, how do prokaryotes do this? Or what's the bacteria version of what I just learned? And that is stuff you could look up for independent work. So here we go. Let's talk about it. Let's talk about eukaryotic cells. And again, some of this should be a review from the previous chapter when we talked about how cells work. But most genes are located inside the nucleus, right? So if it's in the nucleus, first of all, let me make sure this makes sense. When I say most genes are located on chromosomes inside the nucleus, first of all, if it's in the nucleus, then all the genes are located on chromosomes, right? So all the genes inside the nucleus are located in the chromosomes. Now, when I say most, what I mean by that is, and this is going to be less important as far as the exam is concerned. Well, that's right here, this little point right here. Some genes, some of the genetic information, some of the DNA is also found in the mitochondria for all eukaryotes, and for chloroplasts, for things like plants and protists, like algae, right? So again, as far as we're concerned, and for this exam, for this course, this first main bullet point is the most important. This is what we're going to focus on. We're going to focus on those chromosomes that are found in the nucleus. But yes, in the back of your head, just keep in mind that there's also mitochondria and chloroplasts that have their own DNA. As a matter of fact, I mentioned this earlier in that other chapter where we talked about how cells work, and I find this interesting, but for example, sperm do not have mitochondria. Therefore, sperm don't carry mitochondrial DNA. Therefore, all your mitochondria and every single one of your cells came from your mother. So your mitochondrial DNA came directly from your mother. It's not a mix of your mother and your father. It's directly from your mother. And that came directly from her mother, which came directly from her mother. So when you're using DNA for heritage reasons, like to find out your ancestry, that's a very powerful tool because, again, that doesn't get mixed up, right? That's directly from your mom, your mom's mom, your mom's mom's mom, so on and so forth. Now, granted, there are mutations, and we'll talk about that later. But generally speaking, yeah, that's exactly the same chromosomes, exactly the same DNA that your mom has. Another thing to think about. So we're going to talk about in this chapter how these things reproduce, right? And again, we're going to focus on the chromosomes. So when we talk about how our cells reproduce, we will talk a little bit about this, that, and the other thing, but mostly we're going to figure out what the chromosomes do when they reproduce. So that being said, we don't talk about this. How do mitochondria reproduce, and how do chloroplasts reproduce? That's something you can look up for independent work, right? What is it that they do? Also, what is their chromosome situation? Do they have a circular chromosome, like prokaryotes, like bacteria, or do they have chromosomes like we do? All those things you can look up for independent work. Anyway, each chromosome, and again, we're talking about these up here, the ones that we find in the nucleus. Each chromosome contains one long DNA molecule that has thousands of genes, right? So we have multiple chromosomes in each of our cells. We'll talk about that in the next slide. And each one of them is just one long DNA strand, or molecule, I guess you could say, because technically that's two strands put together. We've talked about that before. We'll talk about it again. But yeah, so that's another good question. And this will be a bit easier to talk about later when we talk about how many chromosomes we have. But I just said that there's thousands of genes on our chromosomes. But as we're going to talk about later in this chapter, our chromosomes are different sizes. So some of them are really, really tiny. Some of them are really, really large. And of course, it's everywhere in between. So then the question is, if you want to look it up, what is the average number of genes on a chromosome? Or you can pick a specific chromosome and look that up. Chromosome number 7, according to this source, has this many genes. So you can look that up for independent work if you want. And again, if you're just sending a one-sentence answer, you don't have to worry about copying or plagiarism or writing a paper. You can just send it in an email. I looked it up, and according to this source, the average numbers of genes in a human chromosome is this. And then you just quote the exact source, right? Because you're not getting graded on how long your paper is. So you can just quote your source directly. Anyway, any questions so far? All right, so like I said, like I was hinting at earlier, the chromosome number depends on the species. And the next slide I'm going to show you gives you some examples of that. That's also very important. This also goes back to something I was saying, I think, on Monday, when I got off on a little bit of a tangent about seedless watermelons. So watermelons have a specific number, the watermelon species has a specific number of chromosomes that it should have. And all it is for seedless watermelons is they have extra chromosomes. And because they have extra chromosomes, they are no longer fertile, so they're healthy. They just can't reproduce. And in this sense, when we talk about they can't reproduce, that means they don't have seeds. But anyway, all things you can look up for independent work. And then finally, a word that you should know, genome. I might ask you this word, but you definitely need to know it because I would definitely use it. A genome is a cell's complete complement of DNA. And once again, a nice, easy, independent work topic. Question me, question your textbook. You can send me a paper or even just a sentence, again, just if you just want 1 point. Provide me another definition of the word genome from a different source. Depending on the source, it'll be slightly different. It's going to be probably the same thing, just described in a different way. But yeah, something to consider. Any questions about this slide before I show you a visualization of that bullet point right there? Okay, so here we go. This next slide is very unimportant, but it's a good graphic. It's interesting. I'm going to put a big X to it because I'm certainly not going to ask you any questions about this. Also, just so you know, it's not in your textbook. This is from the old textbook. So if for some reason you were interested in this, you need to download the PowerPoint and look at it. But yeah, there's just some examples, right? Here's the number you do need to know. I should have put it next to the whole thing because obviously we are humans, so you do need to know that we have 46 chromosomes. The rest of those numbers aren't important. I do find it interesting though, like you might figure, the bigger an animal is, the more chromosomes it has, but that's not necessarily true, right? Because look, again, here we have a human with 46 and a giraffe, which is much larger than us, only has 30. Then up here, the smallest, at least on this table, graph, we have a deer with the smallest, right? And then down here, with the largest number, at least on this graph, we have this rat. So obviously size doesn't matter as far as chromosomes. But once again, this should open up the door for many, many independent work topics that are very, very easy. Pick an animal or any organism that you're interested in and just tell me how many chromosome numbers it is. I like dolphins. I looked it up, and according to this source, dolphins have this many chromosomes, right? Also, does this deer have the smallest number of chromosomes for all animals? Does this rat have the largest number of chromosomes for all animals? And also notice this. Yep, just check it. These are all mammals, actually, now that I'm looking at it. So what about the other animals? Right? Are there other animals that have fewer chromosomes? Are there other animals that have more chromosomes? Because remember, animals are a lot more than mammals. We've got lizards, reptiles, amphibians, even things like coral and sponges, right? Even those things are technically animals. So you can look that up for independent work as well. And of course, then we can zoom out even more. That's just animals. What about the other eukaryotes, right? What about fungus? What about fungi? Protists, things like that. Again, just pick an organism. Look up how many chromosomes, what their chromosome number is. Anyway, any questions about this slide? And again, I'm not going to test you. You do not need to know any of those numbers except for the fact that humans have 46 chromosomes. By the time we're done studying this, you should be very familiar with that number because we're going to say it a lot. All right, the next word for attendance, I'm going to circle it. I'm not going to say it just to change things up a little bit. There it is, it's circled. And I'll remind you, if you're online, you have to send those words before 9 a.m. to be considered present. But yeah, that's the next word for attendance. All right, if there are no questions, let's move forward. And this next little bit of information is also going to be a review because you've been introduced to this information before, but here we go. Chromatins are made up of something called chromatin. I'm going to put a question mark next to that indicating I'm not sure if I'm going to ask you that or not. You will definitely need to know the word chromatin because I'm going to use it. You need to know what I'm talking about when I use that word. But I'm not sure there's going to be specifically a question about that on the exam. I think there is on the quiz, but I'm not sure. But yeah, chromatin is what chromosomes are made of. What is chromatin? Let's talk about it. Well, no, let's back up. Sorry. Chromosomes themselves, which is made of chromatin, are usually very thin fibers that are much larger, or excuse me, not larger, but longer than the nucleus that they were put in. Like, for example, I think it was Linnekew or Henson, I forgot, one of you answered that question. How long, if you were to stretch all of our DNA together, how long would it be? And I think the answer was 10 million miles, if I remember correctly. But anyway, so yeah, it's very long. It's just also very thin. So it gets squished together to be put inside the nucleus. Here's something else you should know, though, is the second bullet point. Chromatin is made up of protein and DNA, and the amount is about equal. So there's about the same amount of protein as there is DNA when we look at a chromosome. Usually when people think of chromosomes, they correctly think of DNA, but now I'm telling you there's also proteins. And those proteins are very, very important for biology in general, and slightly important for this semester, because they're going to talk about some concepts later in the semester where those proteins will become important again. So that it's not just a trivial fact that they're there. So for you to understand some stuff I'm going to teach you later, you need to understand that there's also DNA in our chromosomes. Specifically, excuse me, yeah, there is DNA in our chromosomes, but you also need to be aware that there's proteins in our chromosomes. What do? They organize the chromatin. That's one of the things they do, and we'll talk about this later, but the chromatin kind of wraps around, the DNA kind of wraps around those proteins, and I'll give you a visualization of that later. That'll be a little bit more important later. And they also control the activities of the genes. We'll talk about that a little bit later too. So remember, I taught you this, I think, in chapter 2 and or 3. Genes provide instructions on how to build proteins, right? So these proteins, oddly enough, regulate that, right? So these proteins say, all right, it's time now, we need this protein, so let's go ahead and turn this gene on. Or we don't need any more of that protein, so let's turn this gene off. That's what these proteins do, right? They control the activities of the genes, which is obviously very important. And I believe, not the next chapter, but the chapter after that, we're going to talk about the activities of genes. The whole chapter is going to be about how genes work. So obviously, at that point, those proteins will be very important. When fully extended, DNA in a cell is over 6 feet long. And again, if I remember correctly, the person who answered that question about how long all your DNA is, that was one of those things that was considered, right? So you take 6 feet of DNA per cell, multiply that by however many cells we have, and that's how many feet it is. Anyway, I'll put a question mark next to that. That is kind of trivial. Actually, I'll put 2 question marks next to that. Well, a little hesitant to put an X through it, meaning I'm definitely not going to ask, but I'm pretty sure I'm not going to ask you that. As a matter of fact, if I have time, I will go through the exam, and if that question's on there, I'm going to delete it, because that is pretty trivial, right? You can forget that and still understand the concepts I'm going to teach you later. Oh yeah, let me also back up. Later, in a few weeks, we're going to do a DNA lab where you're going to extract DNA. It's all virtual. But one of the things you're going to do, even though it's virtual, it does follow the correct steps as if you were really doing it. One of the things you're going to need to do is put some stuff in there that removes protein from the solution. And this is why, right? If we want to get to the DNA and we're dealing with chromatin, then we need to get the proteins out of there to be left with nothing but DNA. Also, on that note, something to think about. Oh, here we go. Your next word for attendance. This is even better. Instead of giving you a word for attendance, I want you to vote. Do you want to do a virtual DNA extraction, or do you want to do a real DNA extraction where we bring in like some sort of fruit, like maybe strawberries or kiwi, and we put it in a blender, and we mix it with a bunch of stuff, and we chill it, and we move it from place to place, and put some different chemicals in it, and do some things. So that's it. Yeah, vote for that for your next word of attendance. Do you want to do a virtual DNA extraction or a real DNA extraction? So that word for attendance can just be virtual or real, whichever one you want. Or a third option, undecided. If you don't care, then I don't want you to vote. But if you do care, I want your vote to count. Anyway, any questions about that? All right. Here's a little bit more important information as far as this chapter is concerned. This bullet point right here. As cells prepare to divide, the chromosomes coil up. They form a compact structure that can be seen in a microscope. And that's going to be important. You'll see it on the next slide. And again, and the reason I say this is important is because I want to remind you, cells do a lot of stuff when they reproduce. But mostly we're just focused on what the chromosomes do. Just because it just is what it is. So again, as I'm talking about all this, just keep it in the back of your mind, cells are doing other stuff than what I'm telling you. And you can look that other stuff up for independent work. Anyway, so this is as the cells are preparing to divide, right? So the only time you can ever see chromosomes in a picture, or excuse me, a microscope of a cell is when it's preparing to divide or when it is dividing, right? So when it's not dividing and it's not preparing to divide, the chromosomes are all uncoiled and you can't see them. So here's the picture of what I'm talking about. This will be important too, because later in the semester, you're going to do a mitosis and meiosis, actually next week. Yeah, next week, you're going to do a lab where you have to, I'm going to give you a bunch of pictures of cells, and you have to tell me, is it in mitosis or is it not in mitosis? It'll be a little bit more specific than that for analysis, leave it at that. And here, you can see this one is at least prepared to go through mitosis. We know that it is at least prepared to divide because all the chromosomes have coiled up. Of course, it's hard to see these other ones, but here you can see this is a big squiggly mesh, right? You cannot see one particular chromosome in this picture, right? Because they haven't coiled up. They're just loosey-goosey, right? They're just untangled, unraveled. Here, because it's preparing to divide or dividing, and we'll talk about that later, you can see like there's a chromosome, right? Now, don't get me wrong, it might be hard to distinguish, like maybe is this one splitting this way or this way? That might be a little bit hard to tell, but clearly you can see the chromosomes as opposed to these others where it's just a big mess. Any questions about that side? All right, let's kind of back it up and go back to this idea of proteins. So remember, we're talking about chromosomes. Chromosomes are made up of chromatin. And the chromatin is made up of about half DNA and half protein. So let's talk about those proteins. The DNA in the chromatin is packed into an elaborate multi-level system of coiling and folding. And it does this around the proteins. So now I'm going to give you some names of these proteins. We have histones. Those are the proteins used to package the DNA. And then we have nucleosomes. This is the DNA around those proteins. This will make a lot more sense when I show you the picture. Just making sure everything's okay. Also, speaking of which, let me make sure we're still recording. Bear with me, please. I'm sorry. Hey, we are still recording. It's a miracle. An October miracle. Anyway, there we go. So yeah, before I take questions about this, let me just show you a picture of what I'm talking about. So here's the DNA double helix. You're already familiar with that. First of all, I'm sure you've seen it your whole life. Then also we talked about it in chapter 2. We also talked a little bit about it in chapter 3. That's just the new polynucleotide, right? That's just the DNA double helix. That's just a bunch of nucleotides put together. That's what most people are familiar with. But technically, this DNA, again, it wraps around these histones. And technically, when that DNA is wrapped around those 8 histones, not that you need to know that number, that little chunk of it, that little chunk right there is called a nucleosome. So I'm going to put an X through this, which only means that I'm not going to ask you about what a histone is or what a nucleosome is. But you still need to know what it is because I'm going to use those terms in the next few chapters, right? So you need to understand what I'm saying when I talk about histones. And you need to understand what I talk about when I'm talking about nucleosomes. So make sure you know what those are. And again, so you can see that's the first level of organization, right? The first level of organization is that the DNA is being wrapped around those balls, right? So it almost looks like beads on a string. But then it gets even more elaborate. So it's not like just this is your chromosome. No. Instead, that also coils around. And it's a little bit hard to see, right? But so this is what we were looking at earlier. There's beads on a string, but those things also kind of coil up like that to make kind of a thicker rope, if you will. And then even that has its own shape, right? So when you look at it, this is what a chromosome looks like. That's an actual microscope picture. But this is what it looks like. So when it unravels, right, you've got these ropes, and those things kind of unravel into this rope. And those things uncoil into these so-called beads on a string. You don't need to memorize each of those levels of organization. The point here is that chromosomes are very coiled, they're very organized, and that will be important later when we talk about how genes work, how we turn genes on and how we turn genes off. To understand that, you need to understand the structure of the chromosomes, right? And that goes back to something I mentioned since day 1. But there's a big relationship between structure and function in biology. And this is a great example of it. Of course, right now I'm just giving you the structure. Later we'll talk about how that affects the function. And of course, we'll just zoom out one more time and look at this whole picture. So again, this is an actual picture, an actual microscope picture. You can see that's what the DNA looks like. It just looks like a bunch of beads on a string. This is a drawing of what we're seeing in that picture. And of course, again, it coils up and then coils up again, and that's what we're looking at right there. Also, I should point out this picture is from the old textbook. It's not in your new textbook. So if for some reason you would like to look at that picture when you're studying, you need to get that from the PowerPoint. Any questions about this?
The nucleosome is a protein?
Um,
Is that what the second protein is?
so glad you asked. Because not exactly. The histones are the proteins. And when the DNA is wrapped around those 8 histones, we call that a nucleosome. So let me think of a good way to put it. A non-biology example. All right, so I'm sure you've heard of like bacon-wrapped stuffed jalapenos, right? So you got bacon, you got stuffed jalapeno, but when you put them together, it's a bacon-wrapped stuffed jalapeno. So they're separate things, they're separate entities, but when you put them together, that's what they are. So here you got DNA, you got histones. When you put them together, you've got nucleosomes.
8 histones wrapped in DNA is what a nucleosome is?
Yes. And just to remind you, since you're asking, this is a good time to remind you, there'll be no test questions where I ask you what a histone or a nucleosome is. But the only reason you need to know those words is because I will use them in the future. And also, the fact that there's 8 of those histones per nucleosome, the number is also very unimportant for us. The point is, DNA is wrapped around proteins. And that part will be a little bit important later. But again, in my opinion, the names aren't important. Other than later in the semester, when we talk about how genes work, and I say the DNA is wrapped around the histones, you just need to know what I mean when I use that term. Good question. Any other questions? All right. So that's a good background information about duplicating chromosomes, eukaryotic chromosomes. So now let's talk about what we're really here for, which is how chromosomes duplicate, right? So we're gonna talk about how they do that specifically. And then we'll back up and talk about how it happens chronologically, right? So we're gonna talk about how it happens, how the mechanism of DNA duplication happens. Then we'll talk about this whole cell cycle and what happens first, and second, third, and so forth. So here we go. Before a cell divides, obviously the DNA has to be copied, right? So if we're gonna take a cell and basically split it in half so we have two new cells, but the two new cells are identical to the original cell, then obviously we need to double everything in the cell, right? We need to double the amount of DNA, double the organelles, double everything. But once again, in this chapter, we're not focused on everything, we're only focused on the DNA, we're only focused on the chromosomes. So obviously, again, before these cells can reproduce, before they can split in half, which is what this whole chapter is about, the DNA has to be copied. Here's another word that I'm probably not going to ask you, but I'm going to use it a lot. Actually, you definitely need to know this term. Sister chromatids, right? So what happens is you've got a chromosome. When it duplicates itself, or when it becomes duplicated, then the result is something called sister chromatids. This will make a lot more sense when I show you the picture, but let's try to imagine it with words first. So again, you've got this one chromosome. I'm going to try to draw it. It'll make more sense when you see a picture of it. So we got this one chromosome right here. Then it duplicates itself, and what happens is this gives you that classic look that you're probably used to seeing from a chromosome, right? It looks like that because it has duplicated itself. So now this one chromosome has just turned into two sister chromatids, right? There's one here on the left and one there on the right. That's what a sister chromatid is. That's very important because I'm definitely going to use that word a lot. And also, when we start talking about the differences between mitosis and meiosis, this is one of the big differences, and I'll tell you why when we get there. But for now, let's just know that that's a very important term because it'll be important in distinguishing between mitosis and meiosis. And a lot of your exam, or a good part of your exam, will be about differentiating between the two. So anyway, that's what a sister chromatid is in general. Let's get a little bit more specific. They contain identical genes because, again, remember I had that one chromosome and then it copied itself. And in theory, that is a perfect copy of the original chromosome. I said in theory because there are mistakes that happen, and we'll talk about those mistakes later in this chapter. But as far as we're concerned, academically, as far as the exam is concerned, it's just a perfect copy. Also, just so you know, they are joined together at the waist in a place called the centromere. That's what I drew right there. That's another word that you need to know because I'm going to use it a lot when I'm talking about mitosis and meiosis. A lot of things happen at the centromere, so for that reason, you need to know what I mean when I talk about it. And the next slide I'm going to show you is a better picture of what I tried to draw. But are there any questions so far? All right. So here's what I'm talking about. A better drawing. So there we have the original chromosome, right? But it's ready to go through asexual reproduction. It's ready to split the cell in half. But before it can do that, it has to duplicate itself. And that's what it did, right? So we went from one chromosome to two sister chromatids that are joined together at a centromere. And I will point out, just so you know, not that this is necessarily important, but this is just like a generic picture of a chromosome. Each chromosome looks different. Some of them are really big, some of them are really tiny. Also, if I'm drawing it like this, some of them might have like centromere way up here. Some of them might have centromere directly in the middle, like I tried to draw on the previous slide, right? So it's different. There's also, and we don't do this in this class, but in harder biology classes, we actually do factor in, like, where is the centromere? Like, part of identifying which chromosome is which is by looking at the centromere position. But again, for our class, that's not important. I just want to point that out. Just because when you see pictures of chromosomes, which you will in this course, I don't want you to think, wait a minute, that looks nothing like this. So the reason is because this is just a generic drawing of the generic chromosomes. But in all actuality, they look very different. Anyway, any questions about that side? So again, this is how the chromosomes prepare for cellular division, right? They have to double themselves. They have to copy the chromosomes. So what happens after that? When the cells divide, as you might imagine, those sister chromatids separate. And once they are separated, they are then considered regular old chromosomes again. So again, we start with a chromosome, it duplicates itself, and then we have two sister chromatids that are attached to each other, perfect copies of each other. And then the process of cellular division rips them apart. And once they are apart, then they're just called chromosomes again. And that's what this slide shows right here. It's just an addition to what we've already looked at. So from here up is the picture we already saw, right? You had the original chromosome, it duplicates itself. Now we have two sister chromosomes attached to each other at the centromere. Then they go through cellular division and they get separated. At that point, they're just considered chromosomes. But just like when they were sister chromatids, they are perfect copies of each other, right? So the same genes that are on this chromosome are on this chromosome. Not only the same genes, but more importantly, and this will make more sense 2 chapters from now, not only is it the same genes, but it's the same version of the genes. So the only reason you and I are different, it's not because we have different genes. Most likely, if we were to look at all of our genomes, we probably have almost exactly the same identical genes, but we have different versions of any particular gene. And that's what makes us different. And that'll be important later in the semester. So any questions about how chromosomes duplicate? Good. You understand the basics of it. Here in a second, we're going to get a little bit deeper, and there's some more stuff you need to learn about how that happens. Unfortunately, that was just too easy for a university course or a college course, right? So we'll get into more details later, but for now, that's a good introduction. So the next thing we'll talk about is the cell cycle. So once again, we've just kind of been looking at specific little things, and now we're going to kind of step back and look at the big picture. The cell cycle, which is basically, as you should think of it as the life cycle of the cell, right? So when you think of humans, you think of a life cycle, you think of birth to death, right? From baby to old person, if you're lucky. But cell cycle is a little bit different. In a sense, it is the whole life of the cell, but you'll see how it's different. Let's talk about it. The cell cycle is the order sequence of events of a cell's life, just like for humans, right? You start when the egg meets the sperm, and you are a zygote. And then that zygote becomes an embryo, and that embryo becomes a fetus, which becomes a baby, which becomes a toddler, which becomes a child, so on and so forth, right? That's for humans. So the cell also has an ordered sequence of events that we're going to talk about. But again, for humans, when we think of, or for a lot of organisms, when we think of a life cycle, we think of birth to death, right? But in this case, it's not necessarily birth to death, because in this case, the way we're describing it, nothing is born per se. It does come into existence, but in this discussion, nothing dies. Obviously, cells do die, but not in this discussion. So in a sense, instead of saying birth to death, it is birth and parentheses, because obviously a cell is not going to be born to the point that it's reproduced. More importantly, for the exam, you definitely need to know this. There are 2 parts to the cell cycle. There's the interphase and the mitotic phase. And again, it's a cycle. So in your brain, you should be thinking of a big circle, right? And arbitrarily, I'm going to put the bottom here where it starts and ends, but it's a circle. You can put it anywhere. So I'm saying this spot right here, this is birth, right? In parentheses. This is also reproduction in parentheses, right? It all happens right there. But anyway, the whole cycle, again, is broken down into 2 parts, interphase and mitotic phase. You definitely need to know that. When you're studying, if I were you, I would memorize that before I memorize anything else that I'm about to teach you. Because what you're about to learn is that interphase is also broken down into smaller parts. And the mitotic phase is also broken down into smaller parts. And we're going to talk about what happens in interphase. And we're going to talk about what happens during mitotic phase. So again, we're going to go into more detail, and in my opinion, the best way to learn it is first memorize the fact that the cell cycle is broken down into those 2 phases, then start adding the details. But yeah, 2 phases, interphase and mitotic phase. Sort of like a day, right? You might say a day on Earth is broken down into nighttime and daytime. But then you could get more specific and say, okay, daytime starts with sunrise and ends at sunset. And in the middle, we have high noon or whatever, right? You could subdivide it even more. Same thing here. The whole thing is broken down into 2 parts. And as you're going to see later, we're going to separate those into smaller parts. So are there any questions about the interphase and the mitotic phase so far? All right, actually, one more thing to help, I'm gonna point out before we move forward. Hope that this will help you remember. So again, what we're focused on in this chapter, even though we're gonna talk about the whole cycle, what we're really focused on is this part right here, which is the part about reproduction. Mitotic phase is all about how the cells reproduce. So that's what we're gonna focus on. So luckily, that might be able to help you remember that the other part is called interphase, because that's the part that's in between, right? The interphase is the part that's in between the part that we're studying. But anyway, here's a picture of what I'm talking about. I don't remember if this picture's from your textbook or the old textbook. But if you look at the new textbook, the picture's different, and you happen to like this picture more, then look at, study from this picture instead. But there we go, here's the whole thing. Interphase, right here in the light blue, and then mitotic phase in the darker blue. And once again, like I said in this last chapter, or this last slide, those things are also broken down into different stages. So you can see here that interphase is broken down into G1, S phase, and G2, which we're going to talk about here shortly. And then we have the mitotic phase, which is broken down into mitosis and cytokinesis, right? So even the mitotic phase is broken down into two parts. And we're going to talk about those here in the future. And to make it even trickier, so G1 ends, I don't want to say abruptly, but there's a definite distinction between G1 and then S phase. And then there's definitely a distinction between S phase and then G2. And there's definitely a distinction between G2 and then the mitotic phase. Where it gets a little bit trickier, and again, we're going to talk about this later in this chapter. The mitosis and cytokinesis, again, both two parts of the mitotic phase, those kind of overlap. So it's not like mitosis ends and then cytokinesis begins. They kind of overlap, and we'll talk about that when we get to that portion of this chapter. But are there any questions so far? All right, so there's a lot of information on here that you don't need to know yet. You might be wondering, what is G1? What is S phase? What is G2? And that's the stuff we're going to explain. Of course, these pictures you should be familiar with, right? So obviously, whatever G1 is, we just have a single chromosome throughout G1. Then we go into S phase with one single chromosome, and we end with two. So obviously, S phase is when the chromosomes duplicate. Then we can see these two chromosomes, sister chromatids, come all the way through G2. And then those two sister chromatids enter the mitotic phase. So you are familiar with some of this, at least the chromosomes. Here's a number you need to know, maybe. I'm not sure about the number, but you definitely need to know that the cell is usually in interphase. So even though we're focused, this whole chapter is almost entirely about the mitotic phase, even though that's what we're focused on, just so you know, most of the time, it's actually in interphase. Right? The only time it's in mitotic phase is when it's dividing. So any questions about this slide before we dig in deeper? All right, so once again, like I said, usually cells are in interphase, at least 90% of the cell cycle. Also, I want to point this out. This is a very generic statement. There's all kinds of different cells. First of all, there's all kinds of different organisms. And then within each organism, there's all kinds of different cells. So that's something else you can look up for independent work. First of all, as usual, like I said, you can fact check me. So I'm telling you that a cell is usually in interphase 90% of the time. Look it up. Send me an email with a one-sentence answer. I looked it up, and according to this source, cells are actually in interphase for 85% of the time, or whatever it may be. Or I looked it up, and you're right. According to this source, it's 90% of the time, whatever it may be. Anyway, that's the easy answer, the easy thing to look into. Another thing to consider is, again, this is just generic information. What if you get more specific? What about a kidney cell versus a skin cell versus a nerve cell, right? So, for example, some cells don't really do good at reproducing. So you'd imagine if they're not reproducing, they're probably not going to be in the mitotic phase very often. Also, what about cancer cells? And we'll talk about that at the end of this chapter, but cancer cells are definitely an exception to almost everything I'm going to teach you, as you'll see later in the chapter. But anyway, what happens in interphase? This is when cells are performing the normal functions, right? So it's only the mitotic phase that they're splitting. Every other time when they're not in the mitotic phase, when they're in interphase, they're just doing whatever job they do. Kidney cells are doing kidney cell things. Brain cells are doing brain cell things. Blood cells are doing blood cell things. They're just doing whatever it is they normally do when they're not in the mitotic phase. When they're in interphase, they're just doing their job, whatever that job may be. Then, as they prepare, right here, as they start getting a little bit closer to the time that they're going to reproduce, when they're going to split themselves in half, then they're going to need to double everything, right? Not just the chromosomes, which is what we focused on, which we already talked about, but everything, right? And again, we're not going to focus on that, but we are at least mentioning it right there in that bullet point. I mentioned it. Everything in the cytoplasm is going to double, right? So all the you're going to double the amount of mitochondria, double the amount of lysosomes, all the organelles that we talked about in that previous chapter, they need to double. Obviously, the cell is going to grow in size because it's going to cut itself in half. So you don't want to have like two tiny little cells, right? So before it cuts itself in half, it gets bigger. But again, these two bullet points that I just circled, as far as the exam is concerned, as far as our studying of this process is concerned, that's a little bit less important. Once again, even though we're talking about the entire phase, once we get to it, we're mostly going to be focused on what happens in the mitotic phase. But yes, just so you know, there's other stuff happening. And again, that's about almost as deep as I'm going to get. We're going to talk a little bit more about DNA replication. But for now, that's it, right? I don't talk about how the mitochondria duplicate. I don't talk about how the other organelles duplicate. So that is something you could look up for independent work if you want. That would be more like a paper than a one-sentence answer, but you could look that up. What else? When I say that it doubles everything in the cytoplasm, that's a whole paper. How does that happen? What doubles? How does it double? When does it double? You know, I've already told you when the chromosomes duplicate. I haven't told you when all that other stuff happens, so you can look that up if you want. With that out of the way, let's dig deeper into interphase and talk about G1 and S and G2. I'll go ahead and tell you now, once again, I'm mostly focused on the mitotic phase. So even though I'm covering this stuff, it's only because the textbook covered it, and I'm not quite sure if I'm going to ask you any questions about it on the exam time. But here we go. The first one, G1. G stands for the gap. So it's the gap between the mitotic phase and the S phase, right? So even whoever named the cell cycle, even they were focused on this idea of cellular replication, because the only thing that really has a name is S phase. And we're going to talk about that after this, but S phase is when the chromosomes duplicate. But that's only in preparation for the mitotic phase. So really, when you think about it, whoever named the cell cycle and gave them the different names, they're focused on duplication and preparation for duplication, right? Everything else is just like, oh yeah, that's the gap. That's the gap in between the stuff that we're focused on.
You said the gap between mitotic and.
Good. I'm glad you asked that. No, G1 is part of interphase. So what we're looking at, like the previous slides, we talked about interphase in general. Now we're zooming in and talking about the three different parts of interphase. And the first part of interphase is G1. If anything, and again, I'm going to give you some information because you booked it, but if anything, you would just need to know the chronological order. So it goes G1 comes before S phase, and then of course we'll talk about the other stuff later. But you need to know that G1 comes after the mitotic phase and before the S phase, and that it is part of interphase. So again, the whole cell cycle is made up of interphase and the mitotic phase. We are now zooming in, so to speak, and we're looking at interphase more deeply. We're looking at the three different parts of interphase. The first part we're talking about is G1. At this point, each chromosome is single. In a sense, you do need to know that, though that won't be a test question. But when I say in a sense you need to know that, what I mean is you definitely need to know, and I'm going to teach you this in the next slide, S phase is when the chromosomes duplicate. You do need to know that. So by knowing that, logically, then you would also know that before it does that, the chromosomes are still single. They haven't duplicated. Like I've already said earlier, for the entire interphase, generally speaking, the cells are performing whatever it is they do. You know, so kidney cells doing kidney cell things, skin cell doing skin cell things. They're just performing as normal. And what do I mean by normal? Well, they're just growing, right? They're growing, they're accumulating material, they're accumulating energy, they're just doing their thing. But again, for the exam, I would say these are the most important bullet points. G1 is just a part of interphase. It's the part that comes before S phase. And that is where we will end. The last word for attendance will be energy. So see you guys at 10 o'clock for that. Don't forget closed-toed shoes and long pants. And if you're online, don't forget to send those attendance words before 9 a.m. to be considered present. I have to turn in attendance soon, so now's the time to get caught up on your absence.